Hello and welcome to everyone to yet another MedChem Express webinar series. And I'm just taking some time here. It's just 1 minute past 3, so I'll allow for all the registrants to join in first. I can see the number is still going up, so I'll take my time and allow everyone to join in. Okay. I can see that the number of participants are still, increasing, but without taking too much time in the introduction, I think I'll just go ahead and start off the webinar. So welcome everyone, and I think you're prepared to learn something new today, to learn something exciting in the space of immunology. I am Ankita Podal. I am the sales manager looking after the Australia New Zealand region at MedChem Express, and I'll be your host for today. And it is my absolute pleasure to introduce Professor Ming Man from John Curtin School of Medical Research at ANU, who is also a CSO, Centenary Fellow, and NHMRC Leadership Fellow at the university. And before I begin, I'd like to extend a huge congratulations to Professor Mann for the fresh new announcement from Clarivate announcing him as as the highly cited researcher for 2024. And it is awarded for producing multiple highly cited papers in the last decade that rank in the top 1% by citations for field and year in the Web of Science. And he's won it in 2020, 2022, 23, and yet again in 2024. This is just one of the many accolades he has achieved. And talking a little bit about his academic background, he got his bachelor's and master's degree from UNSW. He did his PhD from the University of Cambridge. And then he moved on to the prestigious St. Jude's Children's Research Hospital to get his postdoctoral training, after which he joined Australian National University as a group leader, where he's a professor now. And it is particularly exciting for me to talk about someone and to hear from someone who has achieved who many would think an impossible feat. Ming Man became a full-time professor at a remarkable age of 33. And this is something, what dreams are made of, or something so out of reach, but he's been able to make it is reality. And this accomplishment speaks volumes of his not only of his hard work and dedication, but also of his passion for learning and teaching. And talking about his research background, his lab focuses on innate immune activation in the host defense against infectious diseases and toxins, and how uncontrolled inflammation can lead to the development of cancer. His research has been recognized by the Australian Academy of Science Gold Sack Medal in 2023, the Frank Fenner Prize for Life Science of the Year, Prime Minister's Prizes for Science in 2022, and the Commonwealth Health Minister's Medal for Excellence in Health and Medical Research in 2019. The topic of his talk today is inflammation in infection and cancer, new biological insights and emerging therapeutics. Before I hand it over to him, I just wanted to quickly mention that the presentation time is around 40 minutes, and I encourage everyone to type in your questions by using the Q&A feature, and we'll try to address as many as possible towards the end of the webinar. So please join me in welcoming again the wonderful swimming Man. Just over to you, Professor Man.
Thank you so much, Ankita, for this wonderful introduction. And I'm so delighted to be invited by MedChem Express to give my seminar here today. And of course, welcome to everyone for joining me and the organizing team today for this particular seminar. So I've just shared my screen and hopefully it is working for all of you. So today my talk is about inflammation in both the context of infection and cancer, and the new biological insights from my lab, including the latest studies we have generated and how that informs therapies. So, here's my pointer. So the central question that really drive our lab is how does the immune system actually go about recognizing and also responding, therefore controlling infectious disease and cancer. And so you might ask why you're studying infection and cancer. The two diseases seems totally different. However, there are actually conceptual parallel between the two. So take infection, for example. The immune system can recognize infectious agent like viruses or bacteria through the presence of pathogen-associated molecular patterns, or PAMs. And so these are molecules that make up the structural architecture of any microbes. Take a bacterial cell, for example, here. PAMs are anything ranging from flagellum molecules to bacterial DNA and RNA, cell wall components, or type 3 secretion systems, and bacterial toxins. And so the immune system is incredibly adept to recognizing these PAMs and triggering an immune response. But what about cancer, on the other hand? We know that a stressed cell or dying cells can release danger signals in the form of damage-associated molecular patterns. So what are these danger signals? For example, mislocalized nuclear or mitochondrial DNA, which leak out from their original location into the cytoplasm of the cell or outside the cell, are considered danger signals because they're not supposed to be out there. Similarly, ATP or reactive oxygen species or the influx or efflux of different types of ions can be interpreted by our immune system as danger signals. And so the immune system can equally respond to both PAMs and DAMs and trigger different immune responses and pathways. And the commonality between these two is that we have germline encoded receptors called pattern recognition receptors that can recognize both PAMs and DAMs, and there are overlaps between those receptors. And what we also try to do in the lab is to identify new drugs that can target these receptors or their signaling pathway, allowing us to potentially treat diseases in both of these areas. And so let's take a classical example of how these receptors actually work. So when a pathogen approaches a cell, for example, these pathogens are immediately recognized by toll-like receptors. For example, LPS found on the surface of bacteria can rapidly activate toll-like receptor 4, and that can trigger an immune response. But we know that pathogens are very clever because they can invade and infect the cells, and they can live in this endosomal vacuum where they can replicate to a higher number. But we also have endosomal toll-like receptors that can equally capable of picking these pathogens up, detecting them, and immediately induce this rapid transcriptional upregulation of inflammatory cytokines like TNF or IL-6, but also type 1 interferon responses that can induce antimicrobial signaling. So we know that pathogens can also breach this particular barrier and enter our cytoplasm, where they have more access to space and nutrients for further replication and taking over the cell. In this case, we also have several families of receptors in our cells that can pick up these cytoplasmic pathogens, including the NOT-like receptors, NLRs, the AIM2-like receptors, the ALRs, RIG-I-like receptors, and cGAS-like receptors. They are equally capable of inducing this very rapid transcriptional upregulation of inflammatory cytokines and type 1 interferons. What's interesting about these intracellular or cytosolic receptors is that certain members of them from the NLR and ALR families can form these giant multi-protein complexes called inflammasomes and panoptosome complexes. And these serve as an additional line of defense against the pathogen because they can trigger more inflammation and lead to an explosive cell death called pyroptosis or panoptosis, eventually killing the cell. And so what do they look like? Well, this is a healthy macrophages under just bright field microscopy. They're nice and round and glossy looking. They have these membrane folds, as you would expect from immune macrophages that can pick up different molecules from the environment. But once they undergo this inflammasome or panoptosome-induced cell death, they kind of swell up into like a balloon shape where the molecules from within get leaked outside the cell. They have a condensed nucleus, and eventually they literally explode, rupturing the membrane, as you can see here. The whole membrane is kind of like lifted off and then expose the intracellular components, and all of them basically get leaked out. So you might ask, why do the immune system and the host need this kind of inflammatory, violent lytic cell death? Well, I think an example I can give is if my apartment or home is infested by just one insect or cockroach, I can deal with that cockroach like one bacterium easily with my shoes, right? I can whack it and it's gone. But if my apartment or home is infested with 10 cockroaches running around my bedroom, then I would run to my local stores to buy an insecticide or some sort of spray to go home and spray and get rid of these pathogens or cockroaches, right? What happened in this case is that if my apartment is infested with hundreds and thousands of cockroaches, there's no way I can use a shoe or a insect spray to get rid of them, just like a bacteria replicating everywhere inside the cytoplasm. The only option I have is run out of my apartment and just light up my whole apartment on fire, and there's this explosion happening in my apartment. And then what happened is then my furnitures, everything is out on the street, and my neighboring cells can then pick up those signals and say, okay, so he's been infested. Now we use our shoe or insect spray to just spray the remaining dead cockroaches around that might be still alive. So that's one of the ways you kind of understand why they have this kind of dramatic cell death. So how are they initiated, right? So the inflammasome kind of cell death initiated by a whole panel of these immune receptors, right? You can kind of imagine some of them are very specific, like AIM2, which recognizes double-stranded DNA. But some of them are highly non-specific because they can recognize a whole range of PAMs and DAMs, like NLRP3. So once they recognize this activator, then they kind of assemble like a jigsaw puzzle where the receptor then recruit an adaptive proteins or scaffold protein called ASC, which can then recruit the cysteine protease caspase-1 into this same complex, providing a full architecture of an inflammasome complex that can then go on to induce inflammation and cell death. How do they do it? Well, this protein caspase-1 is like molecular scissors, in which it can then proteolytically cleave two very important cytokines called pro-IL-1 beta and pro-IL-18. Once they are cleaved, they become biologically active, and that they are secreted to the outside of the cell to induce inflammation and initiation of adaptive immunity. Caspase-1 can also cleave another substrate called gasdermin D, in which the N-terminal domain of gasdermin D can go to the plasma membrane to literally punch holes, eventually causing that explosive cell death and killing the cell eventually. And because this inflammasome can send so many different types of signals, it is critical in the development of infectious diseases, autoinflammation, and cancer. And I'll give you examples in my webinar of all three cases and how this inflammasome or cell death and inflammation responses relates to these different disease types. So one of the questions that I was interested in as an innate immunologist was, how do these receptors that live inside the cell actually recognize the incoming pathogens? We already knew that the pathogen can invade the cell, and then they can breach this barrier to go into the cytoplasm. And at the same time, we knew that some of the DNA receptors or pattern recognition receptors can be activated to drive this inflammation and cell death response. So what was missing for me was how do these receptors, for example, AIM2, recognize DNA? Because if you look at a pathogen, their DNA are normally concealed and sequestered inside like a cell wall or cell membrane. It doesn't really make sense to me that DNA just pop out once they get into the cytoplasm so that the immune system can conveniently go and sense this pathogen. What we have found was that during this infection process, the cell actually upregulate the production of these guanylate binding proteins, or GBPs, and also its neighboring proteins called IRGB10s. And once upregulated, these proteins are very good at locating and mapping and localizing to the pathogen inside the cytoplasm. What do I mean by that? Well, we can look inside a macrophage and we can immunostain these proteins. So in here, we can stain DNA in blue, GBPs in green, IgB10 in red, and the outer layer of the E. coli in this purple color. You can see that the LPS layer actually surrounds the GBPs and IgB10 inside, and this is determined by the super resolution microscopy. So in this particular picture, you can see that the green layer of the LPS surrounding an inner layer of the IRGB10 with the DNA inside, suggesting that these proteins can actually penetrate inside the bacteria. And further analysis of these microbes inside the macrophage revealed that there are pore formations on the surface of these E. coli, leading to potentially their structural integrity, the loss of structural integrity and the cell shape and the intactness of these bacteria. So we think that this process is very important because, number one, following recruitment of these host proteins to the bacteria to that location, it induces the killing of the bacteria by physical rupturing of those of the bacterial outer cell wall and cell membrane. And secondly, this is important because it allows presentation of bacterial ligands to the actual immune system. For example, once the bacteria is ruptured, the normally concealed ligands like DNA, which are hiding inside, is now exposed in the cytoplasm, allowing the immune system to then sense different parts of the bacterium. So it's kind of like chopping over a watermelon, so you can access the juicy fruit inside and making it more tasty for the immune system to recognize. And recently, we have identified that in one of these members called mouse GPP1, there are actually 4 different regions within these proteins which have very high antimicrobial index and high charge. So this indicated to us that these regions may be the so-called antimicrobial killing regions. So we synthesized those 4 regions colored here. They are around 30 amino acids in length, and we put that directly onto the bacteria and see what happened. Only one of these regions, the orange color region in the globular domain of mouse GPP1, was able to induce this direct rupture and bursting open and killing of the bacteria. So here is our control sample with the Francisella novacida bacterium. But as soon as we put the peptide encoded in this orange little bit here, the pathogen just exploded and died. And so we think that this is important because we can then use the immune system as a blueprint or a map to harness the most active fragments of those proteins in order to create new drugs to kill bacteria. And this is important because we know that superbugs are emerging all the time, and they are multi-drug resistant bacteria that are making difficulty for the effectiveness of antibiotics. So by having additional mammalian-inspired peptides to complement existing antibiotics could be a potential option in our fight against multi-drug resistant bacteria. We are now testing these peptides in the lab to see how their efficacy is good or not, and potentially making them more stable. But we knew that in a mouse model, these proteins, the full-length proteins, are incredibly important. So in these particular mouse models of infection, we can see that when we infect a wild-type animal or wild-type mice with Francisella novacida and track them over 12 days, the wild-type mice survive pretty well. So more than 60% of these mice survive the infection. However, if we genetically knock out IRGB10, GBPs, or inflammasome molecules AIM2 and caspase-1, these mice are exquisitely sensitive to the infection, meaning that they succumb to the infection within 5 to 8 days compared to the wild type, showing you how important these proteins are in protecting us against infectious agents. So, and this also highlights the importance of good inflammation in host defense, right? Helping us to fight the infection. But you might ask, well, what about the other side? What about bad inflammation? How does this process contribute to bad inflammation? So recently, we identified several bacteria which can secrete toxins on the outside of the cell. And they never have to go inside the cell to induce inflammation triggered by these cytosolic sensors. What we have found is that bacteria like foodborne pathogen like Bacillus cereus or Clostridium perfringens can secrete multiple toxins, and they can bind to the membrane and form a pore. And these are hemolysin BL or non-hemolytic enterotoxins or lecithinase. And then we can see beautiful example of this under cryo-EM where we treated liposomes with this toxin. For the first time, we can see that there are pores formed on this membrane surface shown here. And we can see that for all of these pore-forming toxins here. Clostridium septicum is an important rare pathogen which usually cause sepsis in patients which are immunocompromised, including cancer patients or patients undergoing surgery. And 80% of the patients infected with this bacterium die from the secretion of this alpha toxin. So it's incredibly potent in killing patients who are immunocompromised. And they already live in our gut microbiota. And so what happened is that we identified that these pore-forming toxin actually attach to this membrane, and the potassium ions within the cell actually escapes through this toxin pore. So what we have is therefore a perturbation in the intracellular homeostasis of the cell. And another receptors called NLRP3, which is different from AIM2, but they belong to the same similar family in activating inflammasome. They do not directly bind to the toxins like DNA binding to that receptor, but instead it is triggered because they can sense some sort of potassium efflux in the cell, driving inflammasome activation, inflammation, and cell death. We also found that some of these toxins do not form pores on the membrane, but they go inside the cells in this lysosomal vacuole, eventually using its enzymatic activity to burst open this lysosome, again causing disruption to homeostasis, potentially through some sort of lysosomal enzymes or cathepsin release that again trigger NLRP3 activation. So this shows how important one single receptors in our cells is in detecting multiple type of pathogens, which are so diverse, right? They're all from different families. They all utilize different toxins and have different mechanisms of causing harm to the cell. But one single receptor is able to detect all of these conserved pathway to drive inflammation and cell death. So why do we care about this? Well, in this case, actually activating this pathway is actually bad for us, because you can see in this particular mouse model where we can mimic sepsis, we inject the mice with a Bacillus cereus intraperitoneally, which means that the bacteria will disseminate across the whole body. And after just 20 hours of the injection, these wild-type mice succumb very fast to the infection. Whereas mice which actually lack NLRP3 or inflammasome components caspase 1 and 11, these mice actually survive better, which means that these wild-type mice which have massive inflammation driven by inflammasome is actually deadly, whereas in mice which doesn't trigger that much inflammation because it doesn't have the mechanism to do it, they actually survive better. So we thought this is really interesting, allowing us to use pharmacological drugs to potentially block this process and see whether wild-type mice can then survive. So in this model, again, we injected the mice with Bacillus cereus, and then we treated the mice with just PBS as a vehicle control, or MCC 950 as a highly specific inhibitor for NLRP3. So you can see that just 2 doses of MCC 950 completely prevent death of these mice in response to this sepsis model driven by Bacillus cereus. So again, this told us directly that pharmacological or therapeutic blockade of these inflammasome molecules or one single receptor in this pathway is actually very helpful because it's protective against deadly bacterial infections. And so because of these positive results, you might ask, well, in addition to infectious disease, what other conditions is this relevant? We know that in human, NLRP3 is usually resting in a resting state, but it becomes active once it can sense some sort of PAMs and DAMs. And we know that in humans, patients which have gain of function mutations in NRP3 actually is very detrimental because they develop things like skin rash, fever, joint pains, headache, conjunctivitis, and these symptoms occur very early on from birth to throughout their whole life. And you can see this an example of a patient with this skin rash, like driven by NRP3 gain of function mutations, and they can range in severity from the least severe to very severe, including death. And so the current treatment for this patient is actually block the inflammasome cytokine IL-1, right? Using an antibody or some sort of blocker. However, the treatment can be very painful, especially requiring daily injection. Imagine injection into the joint area, which is very painful. And then they have a very low half-life as well. So researchers are thinking perhaps instead of blocking IL-1, which is a cytokine for all the inflammasome pathway, but potentially having a more targeted approach of blocking just NRP3 directly using things like MCC950 or similar molecules might be able to help. And so in this beautiful paper published by a colleague of mine, Rebecca Cole, back in 2015 in Nature Medicine, she actually her team generated a mouse which carry a gang of function mutations in NRP3 in these mouse strains. And what happened is that these mice can be born, but they rapidly succumb after just 10 days of birth because of autoinflammation. So what she then did was to treat these mice either with PBS or with MCC950 to see what happened. You can see that daily injection with MCC950 prolonged the life of these mice. As soon as the drug is withdrawn, these mice also succumb to autoinflammation and die. But of course, in the control group just treated with PBS had no effect. So this tells us that inhibition of NRP3 using a small molecule inhibitor also can prevent death in a mouse model with an NRP3 gain of function mutation, just like in human. And so therefore, a lot of clinical trials are ongoing for a range of NRP3 inhibitors. And here's just a very snapshot of a slide put together by my postdoc, showing you multiple companies are developing NRP3 inhibitors for the treatment of a variety of inflammatory diseases, including CAPS, cryopyrin-associated periodic syndrome, I just showed you in the previous slides, but neurological conditions, neuroinflammation, infectious disease as well, and also cancer. So this is an exciting space where we are also developing inhibitors, and hopefully next time when I see you, I can give you an update on that particular front. So other than infectious diseases and autoinflammation, you might ask, can these receptors also control the development of cancer? And the answer is yes, because they also control not just acute inflammation, but also chronic inflammation. And we know that chronic inflammation is a catalyst and driver of cancer development. And that is because, for example, imagine a cell which is hit by some sort of oncogenic assault. We would expect to have some sort of danger signals like DNA or ATP getting mislocalized. And some receptors can pick this up, and instead of driving inflammation, they can help in suppressing or downregulating inflammation, as well as cell proliferation. And we know that in patients which have impaired functions of these receptors, they are more likely to lead to tumorigenesis and cancer development. And so we and others have shown previously that multiple pattern recognition receptors can control inflammation-associated colorectal cancer. So this is a mouse model of bowel cancer where we can treat the mice with multiple rounds of inflammation-inducing agents in their drinking water. And then after 3 months, we will expect them to develop bowel cancer. So this is a wild-type mice undergoing this treatment. And you can see that after 3 months, they have tumors develop in the distal colon, which is the end bits of the large bowel. However, if you compare this with a mouse which lack AIM2, so AIM2 is a DNA sensor that I told you earlier, which can detect bacterial and viral DNA. But in the context of this bowel cancer model, where we're not inducing infection, you can see that AIM2 knockout mice in this model develop massive number of tumors throughout the entire large intestine. And so therefore, this shows the importance of just one pattern recognition receptors in controlling the development of this inflammation-associated colorectal cancer. We also decided to test other DNA receptors in this model. And if we knock out STING, which is another DNA sensor, or or a NRC3, which is also putatively known as a DNA sensor, these mice also develop massive tumorigenesis in their large intestine in both of these cases as well. And this year, my postdoc in our lab published a paper showing that lacking just one copy of another nuclear DNA sensor called Cu70 also leads to massive development of tumorigenesis. In this case, Cu70 in this model doesn't live in the nucleus, but it migrates from within the nucleus into the cytoplasm to form a new complex that can then drive these anti-tumor responses. But what you can see here is that multiple pattern recognition receptors can control inflammation-associated colorectal cancer. But the knowledge gap still exists because I was wondering, what are the biological functions of these receptors or related inflammasome receptors beyond just inflammation? Do they do anything other than just triggering or controlling inflammation? So we decided to tackle this big question by systematically and comprehensively looking at the role of these inflammasome sensors in a genetic model of cancer, which do not require inflammation. And so we settled down on this APC-Min model, which is basically a mutation in the tumor suppressor called APC, which then lead to the host to have the development of sporadic and familial intestinal tumors in both the small bowel and the large bowel. And so in human, this is particularly relevant because 60% of the patients with intestinal cancer have one or more APC mutations. And in the mice, we can recapitulate that effect of human by introducing this nonsense mutation called T2549A, or the Min mutation, into the mouse, and then the mouse do the same thing. Because without any treatment, if they just survive for up to 20 weeks, they spontaneously develop these tumors in the small intestine. So this is a proximal small intestine. That's the bit where closest to the stomach. This is the middle small intestine, and this is the distal small intestine, with this region closest to the large bowel. And this is the large bowel, and you can see that in all of this region, you can see the tumors are present in these APC-Min mice without any treatment or any inducer. So using this particular model, my postdoc Cheng took on this massive task of genetically crossing our inflammasome deficient mice with this APC-Min mice to generate heterozygous mice carrying the APC-Min mutation. And then he did a second cross where we eventually get a matching wild type mice with inflammasome on the APC-Min background, and the inflammasome knockout mice also on the APC-Min background. And they are littermate control, born from the same parents. So looking at this, Cheng then was excited to then look at whether there are differences between mice with inflammasome and mice without different inflammasomes. So looking at mice lacking the AIM2 inflammasome, caspase-11 inflammasome, NRP3 inflammasome, or NRP6 inflammasome, we were very disappointed to see no difference between the inflammasome deficient mice compared to their corresponding littermate wild-type control. However, eventually, Cheng got excited because one candidate emerged, and that's called NRC4. What you can see here is that mice lacking NRC4 have elevated number of tumors in the small intestine compared to the small intestine of a wild-type mice. So let's take a closer look. So in the wild-type mice, we were expecting to see tumors in both the proximal, middle, and distal small intestine. But we also saw the same thing in NRC4, but more of this tumor in all three regions of the proximal, middle, and distal. You can see that quantified nicely here. Of course, all three regions, we saw significantly more tumors in these inflammasome NRC4 deficient mice. We then look at the tissue closely by looking at several proliferation markers. So first, we stain for Ki67 in the small intestine. You can see that Ki67 proliferating cells normally live in the crypt of the small intestine. And then you can see that in the NRC4 knockout, these proliferating cells are throughout the entire layer of the epithelium. And this is quantification here. And again, if we look at the PCNA, another proliferation marker, these proliferation cells are in the crypt area of the small intestine, but in the NRC4 knockout, they are basically throughout the entire layer. And this is quantification here. So this told us that without NRC4, you're just going to get massive proliferation here. So therefore, the function of NRC4 is somehow to inhibit or attenuate the development of tumor in the small gut. And so you might ask, well, what about NRC4 in human, right? Does it actually have any roles? So we look at this previously published database in Nature Genetics, which already profiles single-cell sequencing of human patients which are normal with no tumor, or they have precancerous polyps, or they have colorectal cancer. So by looking at these three tissue type and looking at single-cell population, we then can reanalyze this data set for our favorite gene of NRC4 and see how they're expressing. So first, you can look at immune cells. NRC4 is expressed largely in the macrophages and B cells in the normal intestine. And these patients also have the APC mutation as well. What you can appreciate is that the precancerous polyps have less NRC4 expression, but the colorectal cancer tissue cells even have a lower amount of NRC4 expressed. And you can see that trend right across from other cell type as well. In epithelial cells, again, you can see NRC4 is expressed in multiple cell types, especially in the hormone-producing endocrine cells. And you can see that the expression of NRC4 is diminished as the disease progresses. Also in stromal cells, we can see NRC4 expression in myofibroblasts and endothelial cells as well. And again, you can see NRC4 just disappear as the disease progresses. So from this human single cell data, we can conclude that reduced NRC4 expression happens across multiple cell types in humans during this malignant transformation process. And so how does NRC4 actually work in controlling cancer? We knew from previous studies that NRC4 work together with another sensor called innate, and they can sense flagellin and type 3 secretion system of various bacteria, and that drive inflammasome activation, leading to caspase-1 dependent cleavage of gasdermin D and cytokine processing, leading to secretion of these cytokines. So we thought, let's have a look at the hallmarks of inflammasome to see if it does the same thing. So what we're looking at here is a Western blot analysis of cleavage of caspase-1 and gasdermin D in the intestinal samples of mice with NRC4 and without NRC4. And each lane represents an individual mouse here. So what we're trying to look for here is a cleavage pattern of the P20 cleaved band and P30 cleaved band for gasdermin D here. What you can quickly notice is that there's no difference in the cleavage patterns between the wild type group and the NRC4 knockout group. We also measure cytokines, IL-1 and IL-18, in the small intestine. And again, we saw no difference between the wild type group and the NRC4 knockout group. So this suggests that NRC4 potentially doesn't perform an inflammatory function during this model of tumorigenesis. But to provide unequivocal genetic evidence that this is the case, we cross our APC mean mice with caspase-1 11 double knockout, which means that these mice lack all of the inflammasome in the body, because these are effector proteases of all the inflammasome. And we compare tumor burden in the small intestine. Compared to wild type, there's no difference between the caspase-11 111 double knockout with the wild type mice. So both our biochemical evidence and genetic evidence showed us that NRC4 can actually suppress tumor development independently of its known function of activating inflammasomes. We also wanted to look at whether there are changes in the gut microbiome, because we know how important gut microbiome is in the development of a colorectal cancer or small intestinal cancer. However, profiling the gut microbiome between our wild-type mice and NRC4 knockout mice reveal no difference in species richness or evenness, or the combination of two in Shannon's diversity. So we were confident to conclude that the effect of NRC4 in this model is not determined or driven by differences in gut microbiome compositions. So through a whole range of biochemical assays and using mass spec and mass spec analysis to look at differentially phosphorylated proteins, we eventually identified several proteins which belong to the DNA damage repair pathway. So to confirm this, we then look at our intestine again and just look at this well-established marker of DNA damage response marker called gamma H2AX, which indicates DNA damage response. So in just wild-type mice with and without NRC4, with normal APC expression without the mutation, you can see very relatively little amount of DNA damage occurring in the small intestine. But looking at the APC mean mutant mice, you can see more of this puncta coming up in the small intestine, but even more so in the absence of NRC4 in the presence of APC mean mutation. And this is quantified here. So this immediately told us that NRC4 somehow contribute to this novel function in reducing the accumulation of DNA damage. So how does it do it? Well, analysis of multiple DNA damage repair proteins finally told us that if we look at the small intestine, again, individual lane represent individual mouse, we saw that the wild type group actually had a very high level of phosphorylation of this checkpoint protein in the DNA damage pathway called CHEK1. So you can see phosphorylation in the wild type group, but less phosphorylation of CHEK1 in the NRC4 deficient group. We then wanted to confirm this by extracting stem cells from these gut tissue samples, and then we grow mini guts or small intestinal organoids. And what we then did with these small intestinal organoids was treated with a DNA damaging agent called etoposide. And you can see that within just 30 minutes to an hour, we can see very robust CHEK1 activation in the wild type group, whereas the NRC4 knockout group had less CHEK1 phosphorylation. So therefore, we believe that NRC4 therefore function to induce the phosphorylation and activation of this kinase of the DNA damage pathway called CHEK1. So what's NRC4 got to do with CHEK1? Does it actually directly interact with one another? To answer this, we express NRC4 in hex cells tagged with a GFP tag, and we use immunoprecipitation and antibody to pull down NRC4. And we were able to successfully pull down and detect the NRC4. Then from this lysate, we can then detect whether CHEK1 is also being pulled down at the same time, suggesting potential interaction. However, we did not observe CHEK1 being pulled down along with NRC4. But this is not too surprising because we knew that two kinases act upstream of CHEK1, such as ATR and ATM, that can directly phosphorylate and activate CHEK1. So we then use the same lysate and pull down NRC4 and to try to detect ATR and ATM. And what you can see here is that pulling down of NRC4 resulted in the detection of ATR, but not ATM, positioning NRC4 as a potential binding partner of ATR here. We then did reciprocal co-immunoprecipitation, and instead of pulling down NRC4, we pulled down ATR successfully here, and then we were able to detect NRC4 binding to it as well. To support this overexpression system, we go back to the small intestine and actually pull down endogenous ATR proteins in the gut. And then we were able to detect endogenous NRC4 along with this process, suggesting ATR can interact with NRC4 in both the expression system and in the small intestine. Then finally, we wanted to ask whether NRC4 can form a multi-protein giant complexes just like an inflammasome-like complex, but in this case, in the context of DNA damage response, because there are a lot of proteins that belong to the DNA damage response. So this is a busy slide, but I'll walk you through what we did. So to answer this question, we basically use small intestinal lysate, and we pull down endogenous ATR from this lysate, and we try to detect different proteins. So here, this is a wild type group with 3 different mouse samples, and the NRC4 knockout group with 3 different samples. So when we pull down endogenous ATR, we successfully detected ATR in all of the samples. To confirm that this interaction is true, we were able to detect NRC4 in the wild type group, so confirming our previous experiment of this interaction between NRC4 and ATR. We then saw ATR interacting proteins also being pulled down here in the wild type group, and also this activated protein called ETAA1 being pulled down in the sample. However, in the NRC4 knockout samples, these proteins are also being pulled down equally well. So therefore, we position ATR interacting proteins and ETAA1 as a binding proteins of ATR, but NRC4 has no role in recruiting their interaction into this complex. We then saw this adaptive protein claspin also being pulled down when we pull down ATR. However, in the NRC4 knockout samples, we see relatively less of these claspin protein being pulled down. So therefore, NRC4 may therefore facilitate the recruitment of this adaptor protein claspin into this ATR NRC4 complex. We did not see interaction with other proteins within the DNA damage pathway like TOPP1 or BRIT1. And we also went on to see whether this NRC4 version required phosphorylation. And from the inflammasome perspective, this was interesting because there are contentious views on the function of this phosphorylation in NRC4, whether it is required or not required for inflammasome activation. We can detect phosphorylation of NRC4 in the whole cell lysates of the small intestine, but the version that we pull down for NRC4 is not the phosphorylated version. So still, we do not know what the function of phosphorylation of NRC4 is. So finally, we always wanted to go back to human and see, well, is the pathway you identify in human actually relevant in this context? So to do this, we look at the small intestinal samples from genotype tissue expression portal. And what we can see is that increased expression of NRC4 actually correlates with increased expression of multiple human DNA damage response gene, including ATR, CHEK1, ATR-IP, or claspin. So you can see a positive correlation between expression of NRC4 and these DNA damage pathways. And also, we took the top 500 genes that show the highest correlation with NRC4 and then did pathway analysis. And what you can appreciate is that this pathway analysis revealed that most of these genes belong to either DNA replication, mismatch repair, or cell cycle proteins. So again, in the human database, it's confirming our pathway analysis and data from the cancer model in mice. And so our proposed mechanism for this particular piece of work is that we found a novel function of an inflammasome protein that formed like a scaffold complex in response to genomic stress in the nucleus. And I haven't got time to show you, but the CAR domain of NRC4 interact with the HEAT or the FATC domain of ATR. And this interaction is necessary because it can recruit multiple protein, importantly, the adaptor protein claspin. And we know that claspin is very important because it can then bring in CHEK1 into the same complex, allowing the kinase domain of ATR to phosphorylate and activate CHEK1. We also show that after this, CHEK1 can then phosphorylate a cell cycle protein called CDC25A. And this phosphorylation actually leads to its degradation. So therefore, these cells undergoing damage do not undergo cell cycle intervention. Instead, this process initiate and promote DNA damage response, therefore reducing the amount of damaged DNA and gamma H2AX DNA damage marker. And so therefore, this cell is less likely to become malignant and therefore reduce tumor development. On the other hand, in the absence of NRC4 or reduced expression of NRC4 in humans, we know that this complex is not properly formed, so claspin cannot come in to efficiently recruit CHEK1 to activate this process. So therefore, this cell cycle protein is initiated to initiate cell cycle and cell division instead of focusing on repairing DNA damage response. And therefore, it could lead to genomic instability and enhanced tumorigenesis. So therefore, conceptually, we think it is important and interesting to then focus on how to turn this NRC4 green reaper protein, which normally induces this lytic violent cell death and inflammation, into a more anti-cancer proteins like Bob the Builder, where its role is to provide a scaffold into this anti-tumor complex. And maintaining our health and preventing the development of tumorigenesis. And so this is an example of our work showing how identifying the molecular mechanism and pathway in this response can then potentially inspire new targets so that drugs can be developed to activate or inhibit this pathway in not just cancer, but also in the context of infectious disease and autoinflammation as well. And so with that, I would like to close and thank my lab members who did the work. Cheng Shen is the one that drove the NRC4 cancer project. And then, of course, with the assistance of our lab members, I'd like to thank our collaborators within the Australian National University, within Australia and overseas as well, for their support and intellectual input. I'd like to thank our funding sources, the ANU, CSL, and the Australian government for their support. And thank you very much for joining me for this particular webinar. And I hope you enjoyed it, and I welcome any questions. Thank you.
Thank you for the fantastic presentation, Professor Mann. And I can see that some questions have already been typed in. I'll just go to the first question. I think it refers to one of the first few slides you shared, showing the action of GBPs and IRGB10. Why did you use Francisella novicida as the test organism? And it was asked by Yuji.
Oh, that's a great question. So Francisella is not a well-known pathogen. It does cause it. Francisella novicida is a mouse-adapted pathogen, and Francisella tularensis is a human pathogen which can cause rapid fever and has been linked to potential bioweapons. We used it because it is an excellent pathogen that can invade the cell and go into the cytoplasm. However, there are other pathogens like Listeria can also do the same thing. So in that particular work, we actually use things like Francisella and E. coli as a model organism to study how these proteins actually recruit to this pathogen. And our later work actually show a lot of different pathogens also induce the expression of GBPs and IgB10, and they're equally susceptible to attack by GBPs. And an important point is that what's interesting is that we have several GBPs, and some GBP seems to have preferences for different bacteria. And so that's an interesting direction we are trying to follow up and see why some GBP want to attack some group of pathogens and some GBP want to attack another group of pathogens.
Thank you. I hope that I answered your question. Now, the next question is quite interesting from Raju Gurung from South Korea. And he asks, how do you see the role of PROTAC drugs in the treatment of inflammatory and various other diseases, including cancers, rather than the inhibitors?
That's an interesting question. My research is not focused too much on PROTAC drugs. So I'm not able to answer that particular question of how relevant they are in inhibiting or activating various inflammatory diseases.
Great. I think with the rise of advanced technologies like PROTACs and ADCs that are always like, exciting research ongoing, but the sole focus of your research is on inhibitors. So that's quite understandable. Now I'd like to pick another question that's posed by Selena Patel. Have you studied or about viral infections like COVID as well? Because there is inflammation and cytokine storm. That's one of the main reasons of mortality as well.
Yeah, that's a very interesting question. Of course, viruses, it's all around us, and it's especially COVID and what we've experienced during the pandemic. Viruses are very interesting. So in the context of inflammasome, they are being active. So viruses can trigger a lot of inflammation through the inflammasome pathway, especially the pathway of NRP3, and some of our work on influenza A viruses, for example, shows that another sensor called ZPP1 can recognize some sort of nucleic acids and proteins that can drive NRP3 activation and panoptosome activation. In the context of COVID-19, others have already shown that again, that virus is very well equipped in activating inflammasomes like NRP3 as well. So therefore, a lot of the viruses can also converge and activate this kind of plethora sensor or global sensor PAMs and DAM in the context of NRP3. So it is a very interesting space, but my previous experience have only been really relatively restricted to like flu. But there are a lot of exciting research in this space working on multiple viruses like COVID.
Great. Thank you for that response. I have a question here back from our Shanghai office. And it says on page 22, you've showed that the time to test the protein was set at half an hour to 1 hour. So how did you determine that particular time to test?
So this question relates to the mini gut experiments where we grow up the small intestinal organoid and then treated the organoids with the DNA damage agent called etoposide. So we did a range of time points from half an hour to 1 hour to 2 hours. And actually the earlier responses where we see really robust CHEK1 activation, which suggests that the response to this DNA damage agent was very, very quick. And then after the 1 hour response, we actually see the activation is kind of downregulated. So which means that the cell had potentially already repaired the DNA damage. So therefore, the CHEK1 phosphorylation is diminished. And so that's why we picked a very early response to see this crucial difference.
Sure. One more question from Kali Muthu Kovendan. What are the difficulties of inflammation and in cancer research? I think that's what he means. Or what could be the new biological aspects of the therapeutics? For me, I think therapeutics that would have more ease of translation.
Yeah, you're right, Ankita. So ease of translation is one. And also inflammation, as you've seen from my talk, can be positive and negative. So at which stage do you treat the patients with an anti-inflammatory to prevent cancer? At the same time, you don't want to have prolonged anti-inflammatory drugs in the system, because then the person getting the drug would be more susceptible to a variety of infection, right? Viruses, bacteria, fungal. So it's about kind of finding the right time point and the right amount of time for the treatment that can then stop the initial transformation of the cell without then further compromising the normal immune system that can fight everyday germs that can make us sick. So I think that together with what Ankita has said, ease of translation, how accessibility is the drug, how do we deliver the drug to find the tumor, a variety of issues would be very important for consideration.
Sure. One more question from Selena. Is MCC 950 in clinical trial? Because it seems extremely effective.
MCC 950 is probably the most effective small molecule inhibitor for NLRP3, and it works by directly binding to the Walker A regions of the NAC domains of NLRP3. So previously, even before researchers knew about the precise mechanism of MCC 950 binding and inhibiting NLRP3, it has undergone clinical trials for patients with rheumatoid arthritis. In those very early clinical trials, unfortunately, MCC 950 induced some sort of liver toxicity. So therefore, it was discontinued. And so that's why there's this excitement in modifying or creating new inhibitors that can target NLRP3 to drive this clinical translation. Hopefully, the next candidate will work similarly to MCC 950 without the liver toxicity issue.
Sure. I'd like to sneak in one more question from my side. I can see you've worked on small intestine organoid models. Do you plan to extend this experiment to other models or extend to other various kinds of cancer? And what's your immediate goal or plan?
Yes, we are always interested in multiple cancer types because we really wanted to understand whether the pathway that we're studying is not just restricted to the colon or the small intestine. So we're particularly excited to start looking at liver cancer models and skin cancer model and to see how our molecular pathway can apply in those cases, and how drugs that we develop eventually can also target those tissue and organ in those systems. So I guess hopefully the next time I see you, we'll have new data on cancer types other than small intestine and large intestine.
I think we're almost towards the end of the webinar, which was set to 1 hour, but I can see one interesting question from Madhav. Could parasites also be good test organisms?
Yes, parasites incredible. They can also infect the cells, and it has been shown that inflammasome responses can be initiated, and it has been shown that GPPs and similar molecule can also recruit and attack the parasite within the cell. So very interesting space also in the parasite world for innate immunity, inflammation, and cell death.
Wonderful. I think we're approaching towards the end of our timing for today. We will definitely note your questions and we'll try to send you responses after the webinar. I hope this webinar was really valuable for all of you. And at MC, we are always dedicated to sharing insights and having meaningful conversations around the topics that can really make an impact and in the vast world of research. So stay tuned. Feel free to join our upcoming webinars as well. A huge thank you to Professor Ming Man for his valuable time today, and thank you for all the attendees for joining. We'll see you again.
Thank you so much, Ankita, and thank you everyone for joining me for this webinar.
Thank you. Thank you. Well, have a good day. Bye-bye.